Great. Thank you, everyone, for coming to VanBug's February seminar session. Just a note before we start, anyone that still already hasn't, please mute your mics. You can feel free to unmute when you're asking questions, but during the talks, just leave the stage for the speakers. This event's being recorded, so we'll be uploading this to our YouTube channel afterwards, just like all our other remote and in-person seminars. The schedule today, we'll be finishing up some announcements just at the start here. We'll have an introductory trainee talk for 15 minutes, and then an hour-long featured talk from our keynote speaker. There's going to be some time after each talk dedicated to any questions you guys might have. VanBug is organized and run by organizing committee of volunteers, aided by some faculty members. These are the people that make all this possible, so huge thanks to them. We have a few upcoming events, a March speaker and an in-person event in May on Langara College. We'll be sharing more details for those events very shortly over our mailing list, which you can access on our website. There's a little link on the contact page to sign up for that. Outside of VanBug, we also have the Big Research Day on March 20th. And they're going to be running a large in-person seminar with some keynote speakers, some kind of quick presentations from students and trainees. Notably, one of their keynote speakers is the Nobel laureate, Sir Richard J. Roberts, who won the 1993 prize for physiology or medicine. If you'd like to hear about our events or any other outside events that we share, there's, of course, the mailing list that I mentioned, which you can access on our site. As well, we have a calendar and some social media that's listed here, as well as on our site. If any of you are interested in hearing a speaker present, this can be any researcher that you think has cool things that you've read, or you know that they're a good presenter, or you're just interested in hearing them talk, please feel free on our website, vanbug.org, the contact section. There's a link to submit the names of any speakers that you're interested in. We try our best to involve the audience, so anyone who gets picked will likely come from you guys. We are sponsored by the Graduate Studies in Bioinformatics at UBC and SFU, Genome British Columbia, SFU's Big Data Hub, UBC's Michael Smith Laboratories, Langara College, Stem Cell Technologies, and the ISCB, as well as our sister bugs, Monbug and Torbug. For the audience, I'd just like to ask any questions you have. After the, you can leave them in the chat, and speakers can choose either during or after their talk to answer them, or after the chat, you could also raise your hand and unmute. Our first speaker, our introductory speaker today, will be Parham Kazemi, who is a PhD candidate at the Birol Lab with the University of British Columbia. And his talk today is titled Modeling K-mer Profiles of Sequencing Data with Evolutionary Algorithms. And then our next speaker is Dr. Rob Petro, who is an associate professor with the Department of Computer Science at the University of Maryland, and a member of the University of Maryland's Institute for Advanced Computer Studies and the Center for Bioinformatics and Computational Biology. His main research interests are the design of algorithms, data structures for processing, organizing, indexing, and querying high-throughput genomics data, and in the intersection between efficient algorithms and statistical inference. His talk today is going to be titled The Matrix Has You, the Importance of Accurate, Efficient, and Open Methods Upstream of Single-Cell Count Matrices. Without any further ado, I'd love to invite our first speaker, Parham, up to speak.
All good. Everyone see my slides? Okay. Right. Thank you, Mario, for your introduction. My name is Parham. I'm a PhD candidate in Dr. Raj Bural's lab at BC Cancer's Genome Sciences Center. And I'm here to talk about one of my recent projects, which models K-mer profiles of sequencing data with evolutionary algorithms. I'll briefly start by talking about K-mer profiles. To generate a K-mer profile for our sequencing data, basically we take our sequencing reads, and for each of those reads, we generate all of the overlapping substrings of fixed length, or in other words, our K-mers, and then we count them. Now that we have a list of K-mers and their frequencies, we put these counts on the x-axis and also the number of K-mers that have that specific count on the y-axis, and we have a bar chart that looks like this. Typically, K-mer profiles of sequencing data have a shape that looks like this, and they have different components. We've sequenced, let's consider that we sequenced a diploid genome. Some of the K-mers in our sequencing data, they might not belong to that genome, and they basically usually represent sequencing errors, and they are and they have a distribution that's on the left side of this K-mer profile. Some K-mers come from only one of the two homologs of our diploid genome, and they have a homozygous peak and after the sequencing errors. And then some of the K-mers belong on both homologs, which is represented by the heterozygous peak. And also we have some smaller bumps at the end of the K-mer profile, which are caused by repeats on either of the homologs. Now, our goal here is to separate these components because that helps us gain a lot of insight about the genome we try to sequence and about the sequencing data itself. It also helps us categorize K-mers into errors or heterozygous, and this also allows us to detect K-mers that belong to haplotypes, for example, and it also allows us to build a language model. So the language model basically says that a K-mer, how probable is it to belong to this genome that we sequenced or not. To achieve these goals, we will model our K-mer profile based on a mixture model. Mixture models, basically, we have different mathematical functions for each of those components, and they are represented by a set of parameters. For each of these components, for each of these K-mer frequencies we have on the x-axis, we pass it on to each of these components, our mathematical functions, we get an output, and then we sum this output, and we hope this output is similar to the shape of the histogram that we had in the beginning. Different tools have tried to use different mathematical functions for probability distributions for each of these components. We have, for example, GenomeScope that uses exponential distribution for the errors, or CAP that uses gamma for the errors, and also for the heterozygous and homozygous peaks, we have different tools that use normal distributions, skew-normal, Poisson, and also negative binomial. But regardless of the type of the distribution we use for our components, the aim is eventually we want to find suitable parameters that represent our histogram. This requires us to solve an optimization problem. There are different ways to approach an optimization problem. We have local optimization algorithms like gradient descent, and also other algorithms which just give us the answer based on the histogram shape. The thing is here, the search space is pretty broad, so we can also go for global optimization algorithms instead. And when I was developing this tool, I thought that we can, instead of fixing the type of distribution that we're using for the components, we can try to use different distribution types and see in our histogram which distribution type fits our histogram the best. So to achieve our optimization goal, I used evolutionary algorithms. Basically, evolutionary algorithms, there are two main elements that we have to talk about. The first one is individuals, and each individual in our population of evolution is represented by a vector. And this vector basically shows us the distribution for the errors, the parameters of that distribution, and also the distribution for the heterozygous and homozygous peaks, and the parameters for those two distributions. Each of these individuals, they are evaluated by fitness function, which says that after we've given the value on the x-axis to these components, they give us an output, we sum those outputs, and then we find the difference of those outputs to the original histogram. And we want this to be as close to the original histogram as possible. So we define the fitness as the negative of the sum of absolute differences. In evolution algorithms, basically we start by having a bunch of individuals in the beginning. Each of those may or may not be a good representation for our histogram. And iteratively, we take randomly two of those individuals in our population. We combine them with each other, just like how evolution works. We recombine them by basically taking one part of one individual, taking another part of the other one, and combining them together. And we mutate them. So for example, we take a parameter and then we change it a little bit so it goes to another part of the search space. And then by doing this combination, we have new individuals that have their own fitness values. We put all of these individuals back in our population, and then we have a new, now we have a new population that may or may not have individuals that are more fit than the individuals we had in our previous population. By iteratively improving our population, we can see that we expect that our best model in our population will fit this histogram much better than the one that we had in the beginning. So for example, here we can see that our model in the beginning is not, it doesn't fit the model very well. It's basically initialized with random values, random distributions. And the more we improve our population, the best model is more fitting to the histogram. So this whole method and this whole tool is a part of our toolkit for analyzing K-mer statistics. It's called NT-Stat. It's available on GitHub. And it has also different modules for counting K-mers using counting blue filters, and also counting the number of reads that contain a K-mer, which is similar to the TF-ID of information that we have in natural language processing, and also this histogram analysis module that I just talked about. Right, so to evaluate our algorithm, I designed an experiment. Basically, I took a reference genome and I created a second copy of that reference genome. So because the reference genome is usually haploid, the reference genomes that I took are haploid. For example, I took the T2T assembly CHM13 version 1.1 for a human genome. And also the Drosophila reference genome, which is also a haploid genome. I generated a second copy using Pierce, which is a tool for generating diploid genomes and also simulating Illumina short reads. And I generated sequencing reads using both Pierce for Illumina and ONT reads using our own tool, NanoSem. And I generated the K-mer profiles using NT-Card. And after optimizing and trying to model the histogram that was generated by NT-Card, we can see that our tool is fitting good models to the histograms. And for example, for panel A and panel B, we can see that for different heterozygosity values, but basically heterozygosity is represented by the differences of the two copies that we made. And if the heterozygosity is low or if it's high, we can see that our model is able to detect that. And also for higher coverage, so for example, panel C here, we can see that for higher coverages, these components are pretty separated from each other, and our model is still able to fit our algorithm is still able to fit a model to that. And regardless of if we're generating ONT reads or Illumina reads, we can see that on panel D, it still works for short reads. And we found some interesting patterns while running our algorithm on different data sets. For example, we saw that usually exponential, a combination of an exponential distribution and a negative binomial or Poisson distribution works well with Illumina reads, which is interesting because usually the previous tools that were for modeling histograms, for example, Genoscope and CAT and other approaches, they use an exponential distribution or and a negative binomial or Poisson. But for ONT reads, usually the algorithm finds a gamma distribution for the error probability for the error distribution, and that might be because ONT reads are more erroneous usually, or more erroneous than Illumina reads. And also for high coverage, because both negative binomial and Poisson, their shape parameter, if we increase it to a high value, they converge to a normal distribution. So for high coverage, we can see that our algorithm is fitting a normal distribution for the heterozygous and homozygous peaks. And also for the accuracy of our algorithm, basically we, after fitting the histogram model, we looked at all of the gamers and saw if they if they were heterozygous or if they were erroneous. And we saw that based on the counts, we can see that our algorithm is able to categorize erroneous gamers with this less than 1% error, and it can find the fault coverage of the data sets with less than half fault difference, and also we can find non-erroneous gamers with less than 2% error. Sorry, I had that. And you can also find the ratio of heterozygous gamers with less than 1% error. Yeah, thank you. Right, sorry, this part. Also, I tried this on more experimental data. This was basically some real data sets we had lying around in our computers. We had a human data set, which is the a human data set on ONT, human data set on PacBio, and the genome in a bottle human data set on Illumina, the H002 data set. And we can see that our model is able to find fit models to any kind of sequencing platform that is available. Thank you for listening. I would like to thank all my supervisor, Dr. Enoch Burrell, all of the members of our lab, and also the lab development team. Thanks.
Thank you so much, Parham. Does anyone have any questions in the chat? Okay. Seeing as we're on schedule for the next talk, I think we can invite Dr. Petroa to speak. Thank you again.
All right, can everyone hear me? All right, great. All right, so first, I'd like to thank VanBug for the invitation. Really happy to be here and to be giving this talk. And the purpose of my talk is to try to do two things. First, to convince you that if you're working with single cell data, in fact, if you're working with any genomics data, you should be aware of and perhaps sometimes concerned about how the data was pre-processed. It's something that is often overlooked in many analyses, but is of critical importance. And second, to present some of the tools that we have built in this space and why we think that they're a particularly good choice for doing this kind of analysis. So I'd like to start with acknowledgements. Oftentimes we leave this till the end, but I really want to thank upfront all of the members of my lab because a lot of the work that I'm going to talk about here builds on prior work that was done by other members of the lab earlier. And so, really it's a team effort. Though today, most of the work I'm going to talk about is work that's been done by Dongza, who's a recently graduated member of my lab who now works at Altos, and also work by Noor, who is a senior PhD student in my lab who will be graduating soon. And I'd also like to thank the funders of this work, which is a combination of NSF, NIH, and the Chan Zuckerberg Initiative. So let me start with some motivation. There are a lot of tools for processing single cell data. The plot here is from the Single Cell Tools website. It provides nice visualizations of the different tools that are available for single cell data processing. And as you can see in the bottom right, the number of tools has been increasing quickly over the years. The histogram at the top right shows the distribution of tools to the problem they solve. So the vast majority of tools are solving an array of different problems, with the plurality of tools actually solving problems like visualization, and then many tools for dimensionality reduction and clustering and things like that. And at the same time, these tools have had to become more scalable because the plot on the left is demonstrating the increase in the total number of cells that have been sequenced and reported in studies over time. And it's on a log scale. So the studies individually are getting bigger and there are more of them. And what I would like to draw attention to is, while there's a vast increase in the amount of data available and the amount of tools for processing this data, a relatively small fraction of these tools are actually responsible for doing the fundamental pre-processing of the data to turn the raw sequencing data into a count matrix. So I've highlighted here the tools that are labeled as either dealing with UMIs. If you don't know what that is, I'll introduce that in a few slides. Dealing with alignment of the reads, or solving the problem of quantification. However, good output requires good input, right? So I think the distribution of these tools makes sense because these upstream steps are often seen as pre-processing, and somehow often considered solved problems, even though I'll argue that they're not. And the downstream tools provide ways to do a vast array of different tasks that are much more varied in nature than the pre-processing. So things like predicting or evaluating dynamics, discovering new cell types or cell states, drawing beautiful graphics like UMAPs or vector fields of cell migration. However, standard idiom of computer science is garbage in, garbage out. And it absolutely matters the quality of both the input data and the pre-processing tools that are being used, because if your count matrix is wrong, all of the results that are derived from that count matrix could be suspect. So I want to start out with a couple of motivating examples for why you should care about this, rather than just speaking about it abstractly. So what kinds of difference could pre-processing make? I'm showing here some figures from a paper that I'm in no way involved in, but it was one of my favorite sort of analysis papers that came out last year about pre-processing. So the way that you should view this figure 1 is it is showing over various pre-processing tools or configurations, which are the separate bar plots, how many the mean average expression of cell barcodes that are either specific to a particular tool, as shown in brown, or common amongst multiple tools, as shown in blue. All right. The same thing can be seen for the cell barcodes themselves across different experiments in figure 2, where the blue fraction of the cell barcodes are common amongst multiple tools, but then the brown regions of those plots are unique to a specific tool. And so there's a tremendous degree of variation between which cells are called and the corresponding expression of the genes in those cells that are called. And the variation itself can vary substantially between organism and data set. But here's the kicker. All of the methods here are simply just different versions of the same software, CellRanger, which is the de facto software that is created by 10x Genomics, the same company that creates the kits that are most prevalently used for performing the sequencing experiments. So the version on the left is CellRanger 2. This was an open source tool, sort of the initial public version of CellRanger. In the middle of the plots are 3 through 6, which were sort of closed source variants of the tool. And then recently, 10x Genomics decided to make the source code for CellRanger available, though under a somewhat restrictive license. So you can sort of look at the code, you can only use it on 10x Genomics data, the ability to freely modify it is limited. So you get this kind of variation just amongst versions of the same tool. And here's some more plots from the paper, but I just want to highlight the the overarching theme, which is that the authors state that the choice of the CellRanger version affects scores for quality, average gene expression, clustering results, top cluster marker genes for each data set. And the study indicates demonstrable quantitative effects on downstream analysis from the choice of CellRanger version, which results in widely different Chromium single-cell gene expression data for different CellRanger versions. All right, so in other words, just the choice of the version of the tool can fundamentally affect downstream analysis. And one of the big reasons for this is that in the switch from before CellRanger 6 to after CellRanger 6, there was a change made in how reads are counted as coming from genes, specifically including unspliced transcripts in the total gene count. I'll get to that later. So here's another example from another even more recent paper. And this paper pointed out that a very commonly used tool, Velocito, for estimating RNA velocity doesn't work well when you have a particular set of experiments generated with the 5' sequencing protocol from 10x Genomics. And so the thing I want to draw attention to here in the panel on the left is the counts per cell under different tools, Velocito, Alvin Frey, and CellRanger. Here, Alvin Frey, which is the tool that we've developed, and CellRanger are very concordant, but also in panel C. So in panel C, there is no CellRanger, but this is showing the Pearson correlation between gene expression for spliced versions of the genes compared to CellRanger. And what you can see is that Alvin Frey across the board has quite high Pearson correlation, and Velocito has very low correlation for some subset of data and higher correlation for the last piece of data. And then finally, the figure on the right is showing some results of running RNA velocity and looking at cell state transition graphs. And you can see that the cell state transition graphs are non-trivially different as inferred between these tools, right? And the argument they make here is really that the Velocito tool is not orientation aware, and so it makes a lot of mistakes, particularly which are of the type of taking a read that comes from one gene and incorrectly assigning it in the opposite orientation to a gene that overlaps on the reverse complement strand of the genome. And so this picture here, a figure from the paper, shows the correlation between the counts of one gene under CellRanger, where the orientation is believed to be the correct one, versus the counts of the overlapping gene on the reverse complement strand of the genome, right? So there's very high correlation between the forward gene being correctly assigned in CellRanger and Alvin Frey, and the reverse complement gene being incorrectly assigned in Velocito. And finally, I want to point out that how you pre-process your data can affect even what kinds of questions you ask of the data. So this is a paper that was led by Simone Tabiri in collaboration with other folks from his group and Mark Robinson's group. And I and some of my students also collaborated on this paper. And the key goal of this paper is to evaluate differential regulation using single-cell data sets. So we would like to understand differences in regulation of genes across single-cell data sets. To do this, we have to model the splicing status of genes in different cells. So we need to estimate both the spliced and unspliced count of molecules. And we also want to propagate uncertainty about those counts. And so not every method for generating single-cell expression data can propagate uncertainty. The particular approach here relies on something whose technical details I won't go into, which is outputting splicing-aware counts of transcript equivalence classes. But that was fundamentally enabled by the fact that the upstream tool, in this case, our Alvin Frey tool, can generate these different kinds of output, which enable different types of inference, right? So here, taking the output of the tool, which are counts of these equivalence classes as input, they were able to build a Bayesian model and then to perform inference in this model to determine differential splicing. So the pre-processing choice is important. It affects what kind of results you get from your data, how reproducible those results are, the accuracy and correctness of the fundamental conclusions that you draw, and even what kinds of questions you can answer. And so we've done a lot of work in this space. I'm just showing here some of the different tools we've built in the single-cell RNA-seq space. Very recently, we've also released some tools for single-cell ATAC-seq data. I won't talk too much about those today, but I really want to focus on our single-cell RNA-seq tool, Alvin Fry, and Simple AF, which is a wrapper around it that provides, I think, some nice additional features that are targeted on easing use and increasing reproducibility. And in the process of explaining the tool, I want to highlight some of the challenges that come up when you're pre-processing single-cell RNA-seq data. So let me first describe the Alvin Fry framework and the specific pre-processing challenges that it solves, and why those are computationally interesting. So for people who might not be aware of sort of the basics of tagged end single-cell RNA sequencing, I want to give my one-slide introduction to the cartoon view of how you can think about single-cell RNA-seq. So in general, these methods here, the prototypical example is the 10x Genomics Chromium platform. They are meant for doing high throughput sequencing of many cells. So typical experiments might be many thousands or tens of thousands of cells. So there is a automated microfluidics mechanism for trying to isolate cells. Here, the idea is to try to capture a cell in this isolated bead along with this gem. And the gem is you can think of it like a biochemical reaction chamber that has all of the contents necessary in order to perform the downstream biochemistry, which fundamentally is tagging the individual RNA molecules with some extra data. So within each captured cell, you hope to have one gem and one cell. Each gem has information to tag the molecules with a cell barcode and a UMI. All right, and so these two fundamental things are important for understanding the downstream processing. The cell barcode is a short oligonucleotide sequence that is the same for every molecule tagged by the same gem. So every molecule in the same cell should have the same cell barcode. And that cell barcode is attached to the end of the RNA molecule. And then after the cell barcode is attached a UMI. And the UMI is not the same for every molecule in the same cell. In fact, it's designed to be essentially a short random sequence. And the purpose of these is that I would like to tag every molecule that I'm going to sequence with both a cell barcode and a UMI. Then I would like to sequence all of the reads as normal in a mixture from all of the cells at once. And then in silico, after the fact, I would like to separate out all of the reads that come from a single cell, which I can do by collating the reads that have the same cell barcode. And then I would like to determine within that cell which reads likely come from the same exact molecule. And that can be determined by collapsing them based on the UMI. So in essence, the cell barcode answers the question, which cell did I come from? And the UMI answers the question, which specific pre-amplification molecule did I come from? And the importance here is that because in single cell sequencing, we start with a relatively small amount of genetic data, we have to amplify it a lot. So many rounds of PCR are typically done. And so we can potentially get many duplicate reads from copies of the same molecule, and we'd like to deduplicate them with the UMI. So that's the overview of how the data are generated. So in the case of perfect alignment of the reads to the genes in an unambiguous way, no ambiguity, with no errors in the cell barcode or UMI, this would be a relatively easy question to answer, right? So how would I do this? I could take all of my data. I could associate each read with some gene by virtue of the alignment information, what gene it aligns to. I could then first group all of the sequence reads by their cell barcode. So that tells me for every read which cell it came from. And then I could collapse all the reads that have the same UMI, right? That gets rid of things that are duplicate counts from the same molecule. And then I could count. And so you could imagine in your favorite data frame, library, pandas, polars, whatever, this is a few lines of code, right? And the output is a count matrix where every row is a gene and every column is a cell. And the entry says, how many pieces of evidence, how many unique UMIs did I observe for this gene in that cell? However, in practice, it's nowhere near this easy because both mapping and sequencing are imperfect. We'll have reads that map to multiple genes or multiple transcripts within a gene. The cell barcode and the UMI are subject to errors that happen both during amplification and during sequencing as well. All right, so we have to deal with that in practice. So resolving all the cell, gene, and splicing status origin for fragments is not always simple. Methods need to be smart because fragments can arise from unexpected locations and that need to be accounted for. Barcodes and UMIs can have sequencing errors or amplification errors, just like the biological content of the reads. And reads aren't always clearly attributable to a single gene. So there's mapping ambiguity in the data that needs to be dealt with. Furthermore, the methods that we want to build should be efficient because there's a lot of data. So if I look at what is now a quite old single-cell RNA-seq data set, the PBMC 10K data set, it's sequencing from about 10,000 cells. There's more than 600 million paired end fragments, and the data is 44 gigabytes of raw sequencing reads compressed. And so as the amount of data we process continues to grow and as the data sets themselves get bigger, methods need to scale just to be usable. And we care about scalability in two dimensions. The scalability in terms of time. We want to be able to process the data quickly, especially if we have many threads of computation we can use concurrently. And we like to be scalable in terms of memory. We like to be able to process the data in moderate and stable memory so that the processing memory doesn't sort of scale with the total data set size, and that we can, we can process data on relatively modest machines. So these are some of the challenges. In order to address these challenges, we developed the tool AlvinFry, which allows accurate processing of single-cell sequencing data quickly with a low memory footprint and generating accurate results. One of the things that AlvinFry does, and it was amongst the first tools to process things this way, is that by default it actually outputs not a single count matrix, which is the total count for the number of UMIs attributed to each gene in each cell, but 3 separate count matrices, right? And each one of these count matrices represents not just a gene, but the molecular splicing status of the molecule that the read was originally derived from belonging to that gene. So I'll expand upon this in a little bit, but essentially a count can have one of 3 states. It can come from a spliced version of the RNA molecule from the gene. It can come from an unspliced version of the RNA molecule from the gene, or it could be fundamentally ambiguous. We can't tell whether that the molecule that generated this read was spliced or unspliced. So regardless of what kind of analysis you want to do at the end of the day, AlvinFry produces 3 matrices where the UMI counts are divided by the splicing status. And then the user can aggregate these as they wish. They could add them all together. They could analyze these separately. It provides that kind of flexibility. So the picture here on the left-hand side is the overall framework for the AlvinFry pipeline. I'm going to go through at a high level the different modular components of it. So the first question is, what are we mapping our reads to? What does our annotation look like? So AlvinFry makes what we call an augmented transcriptome reference. The input to this is a genome FASTA file and a GTF file. And the output is going to be an augmented reference that includes both the spliced variants of every transcript from every gene, as well as some representation of the unspliced sequence. So the way to view this is there are 2 different models that we support, and I'll sort of explain what they are. And the fundamental motivation for this, or at least one of the big motivations for this, is to allow people to do this type of analysis called RNA velocity analysis, where by looking at the spliced and unspliced counts of a gene across cells and looking at those ratios, you can try to infer how gene expression is changing in cells over time, or more specifically over pseudotime. So imagine I have a pair of transcripts here, T1 and T2. Every thick box labeled with an E is an exon in these transcripts, and every part of the thin line between them is an intron, here labeled with I. So the spliced transcriptome is just what you get when you splice the exons together. So this gene has 2 transcripts. The 2 spliced sequences are what's shown in the blue box here, the subsequence E1, E2, E3 for one isoform of the gene, and E1, E3 for the second that removes the intermediate exon. The set of introns for this gene are what's shown here at the top now with the exons removed. And so one way to index the unspliced sequence is just to take the union of all of those intronic regions. And so when we build an augmented reference like this, we call this reference the splicey reference. And the name comes from, it is the reference for the spliced sequence plus the intronic sequence, right? So any read that maps to the spliced transcript is going to be attributed to the spliced status of this gene, and anything that maps to the intronic sequence is going to be attributed to the unspliced version of this gene. Now, this is collapsing some information because there are regions of the underlying transcripts that could span these boundaries, right? Something could span the boundary between an exon and an intron. And so you can represent that as well. We support this type of augmented reference called the splice U reference, which instead of taking just the union of the intronic sequences, just takes the full unspliced body of the gene, right? So you index the spliced transcripts, and for every gene, one long unspliced transcript that represents the full length of the gene body. This is not a super important distinction going forward, but I would like you to remember these two different variants because it affects exactly how you count or attribute a read to one status versus another. So that's the thing that we're indexing. We build this augmented reference, and then we would like to map reads to it. So the fundamental problem we're trying to solve in this second modular component of the algorithm is, given a read, can we determine which reference transcript it comes from? And within that reference transcript, whether it's the spliced version or the unspliced version. I'm not going to go into the details of exactly how that mapping is done, except that I'll say, Alvin Frey uses this very space-efficient index that we've termed the PISM index itself, based on the very space-efficient SS hash data structure for representing the k-mers in a de Bruijn graph, and is capable of processing the augmented transcriptome and then indexing it in a quite small amount of memory, 2 to 4 gigabytes, depending on the specific reference you use, and allows mapping the reads generally with around 4 gigabytes of memory usage, regardless of the sample size. So it's a quite fast and memory efficient index for read mapping. The next problem I want to talk about is how you determine and attribute the set of cell barcodes that are present in your data. So once you've mapped the reads, you get this file that we call a RAD file. RAD stands for reduced alignment data. It's just a binary file that contains the mapping information. And from that, we would like to now determine what are the cell barcodes, and if cell barcodes have undergone corruption during sequencing or amplification, can we correct them? That's this next module of the pipeline. So how can barcodes become corrupted, and how might we correct them? So I'm going to talk about just one specific way of processing the barcodes, which is the way that's very common with the Chromium protocol, which is you're given a permit list, which is a superset of all of the barcodes that you could possibly observe in this experiment. You expect that every barcode you observe will have originally come from this permit list or this whitelist. So if I observe a barcode that exactly matches a barcode in the permit list, I know that I've observed reads from this cell. If I observe something that is similar but distinct, it may be that this read was tagged with a cell barcode that was corrupted during sequencing. So in a case like this, I might say that the observed cell barcode is this black thing on the top, observed CP1. The permitted cell barcode is this thing on the bottom, which is all A's. And with a single edit, I could correct the observed cell barcode to the permitted cell barcode. So it's very likely that this read came from actually this cell barcode, right? It should be associated with that cell barcode. That question's easy, but it's not always so simple. So here's a case where it's ambiguous. I have the observed cell barcode 2, but with a single edit, I could correct it either to permitted cell barcode 1 or to permitted cell barcode 2. So how do I determine what to do? Well, the way that we handle this in Alvin Frey is we use a 2-pass algorithm. So first, we're going to scan the list of all of the mapped data and only count barcodes that exactly match something in the permitted list, something that we would expect. And we're going to retain all of the permitted barcodes that have at least some minimum threshold number of observations. The default value we use here is K. In general, the set of observed barcodes is going to be much smaller than the set of all permitted barcodes. And so the possibility of an ambiguous barcode colliding with more than one observed barcode is going to be small. So in the second pass, what we do is we go through all of the data again, looking only at this list of things that we believe to be truly present, those with greater than K exact matches, and then we correct only to this permit list. We can still have ambiguities, and in that case, we ignore the ambiguous reads. We simply discard them. But this table here is giving you an idea of what kind of effect this has. If we didn't do any thresholding, we would be able to recover through this correction process about 2.6 million reads. But if we increase this threshold so that we have fewer collisions, we can recover about double that. But if we increase that threshold further, it doesn't continue to increase. Okay, so this is the basic algorithm. We find what's there, and then we correct things within a small edit distance. There's an interesting question here that is a little bit more theoretical in nature, which is that if you look at the 10x cell barcode permit list and you ask how frequent are ambiguous corrections, it turns out that they're quite frequent in the list that we're giving, right? So the experiment here that we did to explore this was take some true cell barcode from the permit list and mutate it at a single position to something that's not always in the list. And then the question is, what percentage of the time does the new barcode you generated, right, that was one edit away from something in the list, have two or more neighbors that you could correct it back to, right? Have more than one Hamming distance one neighbor in the list. And so what you can see is as you increase the number of barcodes that you consider, that probability of having more than two or more things you can correct to increases quite quickly. And so this means that the cell barcodes are not particularly well designed to be separated in sequence space. I suspect they're well designed to be cheap to produce, but potentially we could do better here if we designed the barcodes themselves better. I'm not going to talk about that in any more detail, but it's something worth thinking about. Okay, so now we have mapped the reads to the genome or the transcriptome. We have corrected the cell barcodes. The next phase of the algorithm is to collate together all of the reads that come from the same cell. And so this process we perform in this bucketed external memory implementation so that we can bound the total working memory of the algorithm. But essentially, the idea is quite simple. We're going to do this scatter gather step. So we have this binary file that tells us all of the read mappings that we have. And we also have this correction map that we obtained from the previous step that says for every barcode, is it present in the true set of cells? And if it's corrupted, what barcode should it be corrected to? So what we're going to do is we're going to go through the original list of mapped reads, and we're going to route each read record to some bucket, which is just a temporary file on disk. And we have a deterministic mapping between barcodes and buckets. So each bucket is going to be belong to one or more barcodes, but it's never going to be too big because we know how many reads come from each cell. And this allows us to partition our problem such that once we have scattered all of the reads into the appropriate buckets, then within each bucket, we can simply load all the reads into memory, group together all the reads that have exactly the same cell barcode, and then concatenate together all these buffers to get a alignment file that is globally collated. So that's what these pictures are showing. The scatter phase puts the reads out to different buckets in the order in which they appear in the original file. Then it collates the reads in the bucket by their cell barcode so that all the reads that have the same cell barcode are adjacent. And then it concatenates everything back together into one globally collated file. The main benefit of this is that now when we want to actually perform quantification in each cell, figure out the gene counts for each gene within a cell, we can process the data cell by cell. We don't have to load all the data into memory at once. We can look at all the read records for the first cell, all the read records for the second cell, et cetera. which allows us to limit our memory usage, and also allows us to parallelize this process, right? We can grab all of the records for the first cell, send a thread off to start solving those problems, grab the record for the second cell at the same time, et cetera. So that's the collation step. You can think of it as a semi-sort. Okay, so the last problem we need to solve is actually counting the UMIs that come from each cell. We call this UMI resolution, and there's many different ways to do this. I'm going to explain one of the ways we do it in Alvin Frye. So the algorithm here that I'm going to describe actually was part of a different paper for a tool we originally wrote called Alvin, which was the predecessor of Alvin Frye. And it takes a parsimony-based strategy to try to resolve the UMIs into the genes they come from. So the goal is we want to characterize the number of distinct pre-PCR molecules that arise from each splicing status of each gene within each cell. But the big challenge is that UMIs can be duplicated, reads can map to multiple different genes, and the UMIs can be corrupted during sequencing. So let me explain, how this problem looks and how we go about solving it. Okay, so what's the problem? The UMI collapsing problem, in theory, is quite simple. So here's a really simple example. I have 3 different sequencing reads. The blue bars denote the UMI. Here, they're all identical, and they all map to gene A. So within one cell, if I see a bunch of reads that all have the same UMI and map to the same gene, I can ask, how many distinct pieces of evidence do I have? Or in other words, how many distinct copies of this gene do I think that I had in my original cell? Well, here the answer is 1, right? All these UMIs are the same. These likely come from PCR duplicates of the same molecule. I should count them as 1 distinct piece of evidence. Here's a related but slightly more challenging problem. Okay, I observed 3 reads that all map to the same gene. The UMIs are almost identical, but in this case, the UMI for the read at the top has a single mutation. It's more likely that this mutation resulted from a sequencing error or a PCR error than that it was a completely different UMI. So I'm going to collapse this down again to 1 unique piece of evidence. It's not too hard to do. If I have many exact copies of the same UMI, it's easy to infer that this one might be an error. But here's a difficult case. I have 4 sequencing reads. They all have similar UMIs, and 2 of them actually have identical UMIs, the uncorrupted blue bar. But the reads map themselves to subsets of more than 1 gene. And this could happen because gene A and gene B are paralogous in the genome, for example. So they have similar sequence. And the dotted lines show the mapping. So here, all 4 reads map to gene A, but the bottom 3 reads map to gene B. And if I ask how many distinct pieces of evidence are there for each gene, well, that kind of depends on how I assign the reads to the genes. If I assign all 4 reads to gene A, I might say there's only 1 piece of evidence. The yellow corrupted version of the UMI and the red corrupted version of the UMI are just sequencing errors. However, if I assign the bottom 3 reads to gene B, there's 1 piece of evidence there, but then there's also a distinct piece of evidence for gene A, because I can't describe that first read as coming from gene B because it doesn't align there, right? So there's a chicken and egg problem. How I resolve the UMIs depends on the assignment of the reads, and the assignment of the reads depends on how I observe the UMIs. So there are many challenges here, right? They stem from the difficulty of cell isolation, small quantities of genetic material, et cetera, but I need to solve this problem computationally. So the way that we solve this problem is we represent the UMIs and the transcripts to which they map as a graph. So the graph that we're going to have is defined by a set of vertices and a set of edges. Here, every vertex is a tuple, which consists of a pair of things. First is a set. That is the set of transcripts to which the read aligns. So if the transcript aligns to a single read, this is just the name of the transcript where the read aligned. If the read aligns to 4 different transcripts, this is just a set of those 4 transcripts. And then the other element of the vertex is the UMI sequence that tags that read. So a given vertex can describe a read by virtue of the transcripts to which it maps and its associated UMI sequence. And if I have multiple reads that map to exactly the same set of transcripts and have exactly the same UMI, then they're equivalent, okay? So I can assign to each vertex a count or a multiplicity. The edges of the graph are drawn between vertices that may potentially be explained by the same pre-PCR molecule, right? So if it's possible that the information contained in two separate vertices could be explained by a single molecule that was sequenced by multiple reads that were potentially corrupted during amplification and sequencing, I'm going to draw an edge between them. And the specific relationships we're going to encode here is for any pair of vertices, if the set of transcripts that label that vertex have a non-null intersection, so there's at least one transcript that could explain both vertices, and the UMIs are the same, then I want to draw a bidirected edge between those vertices. Likewise, if the intersection of the transcript sets is non-null and the UMIs are within a small edit distance, then I want to draw a bidirected edge between them. Finally, if the intersection is non-null and the counts of the vertices are vastly different, specifically if the count of vertex U is at least twice the count of vertex V, then instead I'm going to draw a directed edge. And the reason for this is technical. I won't go into too much detail, but essentially PCR is this sort of geometric amplification process. So if we observe reads where the UMIs are similar, but one is much more prevalent than the other, it's likely that we observed a PCR amplification error rather than a sequencing error, and the directionality of the edge encodes that. So given this graph structure, the thing that we're interested in finding is the minimum number of pre-PCR molecules that explain all of our data, right? So it's a parsimony criterion. In fact, we call this graph that we build the parsimonious UMI graph, or the PUG. And the formal problem is this. I give you one of these graphs, this UMI resolution graph, and I would like to find a minimum cardinality cover by monochromatic arborescences. So that's a mouthful, but really all that means is we would like to find a way to explain all of the vertices in this graph such that we have a cover of the graph by a series of arborescences. That's just the variant of a tree in a directed graph of minimum total cardinality. So each arborescence describes all of the reads that emanate from one pre-PCR molecule, and we would like to explain the whole graph by the minimum number of them. So unfortunately, this problem in the worst case is NP complete. We demonstrated this by reduction from dominating set, but most practical instances seem to be really easy. So we can solve this with a greedy algorithm. So let me give a concrete example. So here is a parsimonious UMI graph. Every vertex is labeled with a transcript set. Here they're pretty simple. The top two vertices are labeled with transcript 1 and 2 respectively, the center vertex with the set transcript 1 and 2, and the bottom vertex with just transcript 1. Here the shape inside each vertex represents the UMI. I haven't drawn out an actual UMI sequence, but the edges are induced by those properties that I talked about before. So I could think about resolving this particular graph into three arborescences. So here's one arborescence, one group of arborescences. It says, I'm going to use one pre-PCR molecule to describe the top left node that's labeled with T1. I'm going to use a second pre-PCR arborescence to describe the set of nodes with the triangle, with the two triangles labeled by T1 and T1, T2. And I'm going to use a third arborescence to describe that bottom node, right? And so here, the idea is that set of intermediate nodes in the yellow cluster here, I can describe with one pre-PCR molecule, saying they both arose from T2. Okay, but this resolution is not optimal. In fact, I could do this instead, because all of the vertices along this left path are labeled with T1. I could, in fact, say there's a arborescence, which consists of these pair of edges that explain all three of these vertices at once. And in that case, they're all reads arising from T1. And then separately, I need one more pre-PCR molecule, which is T2, right? And there's reads that arise from that. So in this case, this is the most parsimonious cover with two arborescences. So in general, given an arbitrary graph, our greedy algorithm tries to cover it with the smallest number of possible arborescences. This is the minimum cardinality one. If we have one or two arborescences, we can guarantee it's globally optimal. Otherwise, the algorithm is just a heuristic. There are cases where the resolution itself is fundamentally ambiguous. So here, there's only one arborescence, but it could be due to either T1 or T2. In our algorithm, what we do is we report this arborescence is actually a cluster that could come from either of these transcripts, and we treat this UMI as ambiguous, and then we resolve it probabilistically after the fact with an EM algorithm. Okay, so I want to note that this problem of performing UMI resolution and modeling it as a graph is a rich way of modeling the problem. We've presented here, or I presented here, one way of solving it, which is what we looked at before. But there are other algorithms one could imagine to try to solve this, and even other ways of trying to model the problem. I think it's a rich kind of underexplored computational problem in single-cell genomics. And in fact, we contributed to this single-cell best practices online book, where we talked in a little bit more detail about this formalism for representing the UMI graph. The original motivation for this is actually due to the underlying algorithm of this tool called UMI Tools, which was developed by Ian Sudbury and Tom Smith, who were co-authors with us on the original Alvin paper. So they sort of introduced this idea of modeling it as a graph problem. We generalized this and came up with a different optimization problem for it, and I think there are probably many more. So I encourage people to think about that if you find these kind of graph problems interesting. So there's other stuff that comes here that I won't have time to talk about. For example, how to assign the splicing status of the UMI. I won't spend too much time there, though we do have work specifically on that. So we had a paper at ISMB last year for this tool called Forseti, which helps better resolve the splicing status in cases where you have ambiguity. So the output of Alvin Fry, after all of these modular stages of the pipeline, is a cell by gene count matrix, actually a cell by 3 gene count matrix, because we have 3 separate matrices, one for the spliced, one for the unspliced, and one for the ambiguous gene states. This unifies the pre-processing because now we can take those count matrices and we can perform any kind of analysis we want. We can sort of add them together and treat them as the total transcriptional output of the gene, do standard single cell or single nucleus analysis. We can keep them separate and look at their ratios and do RNA velocity analysis. So it's a pretty flexible way to process the data. The algorithm itself is quite fast, quite memory frugal, and very accurate. So we did a lot of analysis in the paper where we looked at Alvin Fry, but I'll just give the top level points here. So we processed this data set, which is over 600 million reads. The processing for Alvin Fry with 32 threads took less than 14 minutes from raw reads to final count matrix. It took less than 4 gigabytes of memory using this PySum index, and it gives very accurate results. Here we're looking at non-parametric simulated data from the Star Solo paper, and what you can see if we look just, for example, at one of these columns, like the Spearman correlation, the correlation with the ground truth quantifications is very high. So it's a very fast, very memory frugal, and quite accurate pipeline for pre-processing RNA-seq data. Okay, so pre-processing matters, right? I hope that at least at this point of the talk, I've convinced you of that. It matters how you pre-process your data. But how much do people actually pay attention to this? Because the talk is titled "The Matrix Has You." Why did I call it that? Well, because I think that a lot of people start with a matrix. They may or may not know where it comes from. So a while ago, I was curious about this. So I posted this poll on Twitter, which I refuse to call X. And I got some answers. And the question was, if you work with single cell data, how often do you use a matrix where you don't know the provenance, right? Or rather, I asked, this partial question, you know the provenance completely, you don't know it at all, or you know it partially. And so surprisingly, what a lot of people said was, yeah, oftentimes I work with a count matrix where either I don't know the provenance of it, I don't know what tool generated it, what version of the tool, et cetera, what version of the reference was used for counting. And sometimes I work with a matrix where I know partial provenance. So I know some of this information, but not all of it. I don't think that working with pre-processed data is bad. Sometimes you already have the data processed. You don't want to go back, or you don't want to process it with a new pipeline because you're going to work with other data that was processed in the same way, and you want to keep everything uniform to avoid batch effects. Starting from pre-processed data is okay if you're fully aware of the provenance, so you know how the data was processed and what choices you should make. The constraint that's almost never satisfied in practice is here's the full provenance data, right? Usually what you get is here's the matrix, and if you're lucky, maybe some auxiliary files. And a nice way of thinking about this, someone who responded to my Twitter post was, imagine you're building a structure with steel of unknown provenance. You would really like to know where your building materials are coming from. When your output is a single cell analysis, your building material is the raw matrix. So I want to talk about this by way of analogy to a related tool that we built in the context of bulk RNA sequencing analysis. So this is joint work with several folks led largely by Mike Love, who's a long-term collaborator of mine. And we built this, this tool and this computational framework called TXI Meta. And TXI Meta comes from this amalgamation of transcript abundance import with metadata. And fundamentally, the problem we're trying to solve is very limited in scope. It says, if you process bulk RNA-seq data and you're fundamentally doing expression analysis, so I have quantification results, how can we make it possible to automatically propagate provenance information so that you always know at least the most important metadata about how your data was processed, what version of the reference you were quantifying with respect to, for example. So Mike did, this was before my Twitter poll back in 2017, a Twitter poll that was quite similar. If you're working with public data, how much of a problem is it to obtain just the genome and gene versions that were used when the data was mapped, right? Turns out sometimes you can't determine it. Sometimes you can determine it, but it's a big hassle. Only about 10% of the time do people say it wasn't a problem to figure out exactly what the reference annotation was that was used. And then, he asked, well, if there was some way to automate just the answer to that question, do you think it would be good? And as you would expect, most people said yes. Okay, so here's what a typical bulk RNA-seq analysis might look like. You have some set of RNA-seq reads that you like to quantify. Imagine you use some tool for it. Here, the example is going to be the tool that we've built for RNA-seq quantification called Salmon. So you have some index of the reference transcriptome. You quantify your RNA-seq reads from it, and the output are these quant.sf files, each of which contains information about, for every transcript, what is estimated abundance. And the source of the reference transcriptome could be anything. Maybe you're working in mouse, maybe you're working in human, maybe your reference annotation is from GenCode or Ensembl or RefSeq, right? You just have some set of gene names. The problem comes when you later want to do an analysis with this data. If I give you just the quantification data, and then later I want to, say, merge it with some ChIP-seq data to figure out where peaks occur upstream of expressed genes, I want to do some sort of enrichment analysis, I would really like to use exactly the same annotation. But if I don't know exactly what version of the reference was used with my RNA-seq data, then I can't reliably do the enrichment, right? So I have this problem. So you might ignore the genes in the gene version and just say, oh, well, I have some annotation lying around. It's HG38. I'm going to use that. It could be that this leads to spurious results. You don't find enrichment that you expect to find. Maybe genes or models get dropped arbitrarily from your analysis because they're in one set and they're not in the other. So the details of the data generation matter a lot. You care about what your features are, if they're correctly labeled, who generated the data, what versions of the software were used, which parameters were chosen, all that kind of information. So we built a tool to solve just the problem of what reference did you use in the quantification context. And the way we did that was building this tool called TXI Meta. So the goal was to remove this problem, this red box of missing metadata about the transcript provenance. We would like to give you not just the transcript counts, but some other piece of data that allows you to know with certainty what reference was used when analyzing that data set. So the solution looks like this. So far, everything's the same as in the previous case. But now what we're going to do is we're going to build for the reference transcriptome, when we're constructing the index of it against which we're going to map, a signature. And the signature we use is relatively straightforward. We take a SHA-256 hash of all of the transcripts, and separately a SHA-256 hash of all of the transcript names. And together those form the complete signature of this reference transcriptome. We quantify everything as normal, but then when we create the output quantification results, we package with it that signature of the transcriptome that was used during quantification. Okay, so you have this very small signature that gets carried along with all of the quantification information that specifically and explicitly designates the reference that was used for quantification. Okay, so the way this looks when you actually run TXI meta is you just point it at the quantification directory or the set of quantification directories. It knows exactly what to look for, and it automatically matches the signature to some known transcriptome. So Mike's gone through the trouble of computing these hashes for, basically all of the common references back many years, and every time there's a new release with new versions, we compute and upload the new ones. So here we automatically find out this is a GenCode annotation for Homo sapiens from version 38 of the annotation. There are a lot of other really cool things that come along with this, which is once you know exactly the version that's used, you can automatically link other metadata. So even though you just have the transcript quantification results, we can automatically pull in the gene models or other known annotated features like peaks that were annotated with this version of the reference. Right? So this works out of the box for GenCode, Ensembl, RefSeq, and model organisms. And we also have a way for you to label a specific sort of non-model annotation and then provide that for other users. So if you sort of created your own de novo assembly, you can register that signature and the associated annotation so that can automatically be determined by TXI meta. Okay, so I want to argue that the same kind of idea we used in TXI meta is also very useful in the single cell context. And we don't have a complete analog for that, but we're building one in this tool that we call Simple AF. So Simple AF stands for Simple Alvin Fry. It is a simplified way to run Alvin Fry. When I talked about the pipeline, I talked about all these different components, and Simple AF collapses it down into just two commands. There's one command that indexes your reference, and then there's another command that does the mapping, the barcode correction, the collation, and the quantification all in one step, which we call quant. And it exposes all of the different options that can be passed to Alvin Fry to generate different types of references, to use different types of resolution algorithms, etc. So it breaks this longer pipeline up into just these two fundamental components. But the key context and the key idea is this. Because Simple AF is aware of exactly how Alvin Fry is run, all of the parameters that are used when everything is generated, it is able to propagate that metadata information all the way from the raw input to the final output quantifications, right? So this is this thing that I call an augmented execution context. I don't know if this is a pre-existing term. I coined it a while ago when I came up with this idea. But the idea is we are executing all of these tools in this sandbox where we are aware what the inputs and outputs are when we're doing the generation. And so we can propagate all of the relative information through the steps of the pipeline so that at the end, we can have a nice summary of all of the relevant metadata. So Simple AF is aware of the context and semantics of running Alvin Fry. It propagates important metadata through the different stages of the analysis, and it allows downstream tracking of the provenance of the data to improve reproducibility. The other thing that it provides us is because it's a wrapper around this Alvin Fry tool with all of these different configuration options and parameters, it might be that over time we determine that there are different best practices in exactly how this tool should be run for different types of data. That can be programmatically encoded in Simple AF so that you say, I would like to process single cell data from this protocol, and it automatically sets this set of parameters is likely the best thing for you to use, right? And then that parameter set can also be propagated and recorded in the output so you know exactly how your data was generated. Okay, so some closing thoughts here as I run up on my time. I hope I've convinced you that pre-processing choices are critical in how you do analysis. Often we think of the analysis as starting from a count matrix, but it doesn't. It starts from the raw biological material, and if we're willing to assume that all of the protocol is pretty reproducible and accurate, it at very least starts from the raw reads. The methods that we use for pre-processing should be accurate, transparent, and open. The open part I didn't talk too much about, but I really believe it's important that, fundamentally we use truly open source tools here. If someone comes up with a better algorithm for UMI resolution, I want them to be able to implement it and, publish their own paper on it or upstream it to Alvin Fry. That's not really possible with something like CellRanger. And of course, these tools should be efficient. We don't want to waste computational resources. Openness is important to understand what is happening to our data and to allow us to work towards key standards and better methods. Recently, I've become increasingly involved in scVerse, which is a Python ecosystem for processing of single-cell data, and I'm hoping to really try to push some of the standards and data augmentation ideas there as well. Efficiency is very important. I think this is sometimes overlooked because oftentimes when a new tool comes along or tools get better, we would like to reanalyze existing data. So in the worst case, if you're unsure of the provenance, but you have access to the original data, you can just rerun everything. That's a lot easier if rerunning is fast and memory efficient. Data provenance is critical. I think in some cases, you should not even worry about analyzing a data set if you have no knowledge of its provenance, right? This can lead to all kinds of critical problems, batch effects, and things like that. And so I'd like to close with this call to arms, which is a push for open, transparent, and truly free pre-processing methods, free as in beer and free as in speech. And how we develop and converge on a robust provenance of tracking for single cell data is still an open problem, right? So I think there's a lot of extra stuff that needs to be carried along with the data in the single cell context. And best practices and a community-wide uniform standard for that is something that I think is important to work towards. So with that, I will close my talk, and I'm happy to take any questions that folks might have.
Thank you so much, Rob. That was really good.
Thank you.
I think I had a question. In case anyone else has one, please leave it in the chat. But do you think there's a better way people can communicate their data provenance on like the publishing level? And would that kind of, do you think that could help push for like a stronger focus on healthy provenance?
Yeah, I think that's a great question. I do think that people could do this. For example, something that we try to do when we publish papers, mostly methods papers, is to have some section of the paper that catalogs exactly which versions of each tool were used to generate the data, where possible, the specific command line invocations that were used. I don't think that the publication itself is necessarily the ideal place to put that. I mean, it's good to have it there. What I would really like to see at the least is the deposition in a machine-readable format of the core information along with the data. So rather than just uploading a count matrix to Geo, upload a count matrix along with a JSON file that says, here are all of the commands that were run to generate this data. Here is the output of running -- version on the tools that we used in the pipeline. In order for that to fundamentally be widely applicable, I think we have to automate it as much as possible. But even now, if people, made a best effort attempt to include some metadata along with the data, I think that would go a long way.
Okay, great. I think we have a question from Faraz.
Sure.
Thanks, Rob. It's an amazing talk. So I agree with you that we want to have that JSON file, but what can be done from, let's say, infrastructure point of view, that they need to also enforce that this information should be uploaded alongside your matrix, right? Because you can provide a machinery that generates this data, right? If it's not being enforced to be uploaded, it's not going to be really matter, because people will, they may forget, right? I think there should be some safeguards in place even for that to work. I would say that, we need like we require them to upload, let's say, how biologically sample is prepared, but not how it's been analyzed.
Yes.
So
Yeah. Yeah, no, I completely agree. You know, and I think with any problem like this, there's really two different challenges. There's the technical challenge, which is what metadata do we need exactly? How do we collect it and propagate it as automatically as possible? And then there's the societal problem, right? Which is how do we convince people that this is important? That's part of what this talk was about. But then how do we enforce that people actually use these tools once we've built them? And I agree. I mean, I think that ultimately, like the end goal of where I would hope to get is that you can't upload just a count matrix, right? If we have a minimal viable metadata, you know, package, it doesn't have to be exactly the same for every tool, but maybe there's a minimum subset that should be required with upload of the data. And that way we can at least rely on having this minimum, hopefully machine readable metadata associated with every sample. Now, how to get there is a challenge, but I think that once more tools are available that produce this kind of data, and you can argue for the usefulness of it, then you can go to the people who curate and host the data. You know, we can go to NCBI and folks like that and say, it'd really be useful if your upload forms required this field, right? And even did some minimal validation of this field. But I think that is an equally important problem, but it's a bit more on the social side than purely the technical side.
I want technical question. I was listening and it just came to me. So in the Alvenfry, you have this 2 pass for correction of your UMIs.
Of the barcodes.
Yes, and barcodes. Yep. So the question I have, so basically what what I'm trying to understand, you just basically do the perfect ones, you reduce the whitelist to a smaller subset and then say that, well, since I have seen these ones, most likely everything belongs to variation of this one.
Yes.
Did I understand correctly? So the question now I have, obviously, this works very well with the sequencing errors. But what happens if you have a PCR effect, which is happening in the first cycles? And obviously, like you have 17, 18 cycles of PCR, and you subsample from all the descendants, all the leaves at the end. So this basically the first, like you will have this molecule, and let's say 20 of these molecules, all of them have this base change. It is not in the whitelist, right? Because it's not perfectly aligned.
Right.
But in your 2 passes, you're not going to pick it up either, so it's going to be eliminated. Am I correct in understanding this?
So it's certainly possible for that to happen. You know, however, what I would say is that problem is worse or more pronounced with the UMI than with the barcode. And the reason for that is that if you have a PCR error in this molecule in like say the first round, then sure, the barcode and potentially the UMI might be corrupted, but there would potentially and likely be many other molecules from this cell. So unless all of them had, you know, that same PCR amplification error, then you would expect to see that most of the other molecules from the cell would match the original barcode.
Yeah. Okay. And one question regarding the Alvin, so you had this heuristics, the greedy approach, which
Yes.
is based on the parsimonious approach. So you mentioned that you're using greedy approach. Did you do any analysis bonds? Like I know that you said when you have 1 or 2, it's optimal, but what if it's more than that? Did you do any analysis on lower bonds? Like what are the
right. So great question. So let me, I went very quickly. Let me explain the algorithm very briefly. So it's essentially, we start a walk at every vertex labeled with every transcript, and we find the biggest component we can. Whatever the globally biggest component is, we remove all of those vertices from the graph and we repeat. This is why it's a greedy algorithm. We did not prove any approximation bound for this algorithm. So it's unclear if this is purely a heuristic or if this guarantees like 2-opt or something like that. What we did do is we observed that in real data, in greater than 80% of the cases, we were able to resolve all the graphs down into 1 or 2 components, in which case we guarantee optimality. So we have some empirical bound, but we don't have any theoretical bound on how bad the greedy approximation might be.
Okay, that's great. So I'll leave. I mean, it's always great to listen to your talks and have more questions, but I'll leave the questioning for our trainees as well, so they can ask their questions from your talk.
Sure, thank you very much.
Thank you.
We have one question from Brahman. How to avoid confusion in distinguishing between cells that have similar UMIs?
Okay, right. So here, I guess we mean barcodes, because the barcodes are the things that label the cells rather than the UMIs. The fundamental approach that we take here is a conservative one, right? Which is that if you have this ambiguous correction, and even after running this 2-pass algorithm, you can't uniquely assign the read to a cell based on the barcode, then we simply drop the read. This is a conservative approach because we're saying that we would rather ignore the read entirely than falsely correct it to the wrong cell. And the reason for that is that correcting this read to the wrong cell can then also impact the UMI resolution for that cell, because this barcode is associated with some UMI. There might be other UMIs that are similar to it, etc. So it's designed to be a very efficient algorithm, and it fundamentally sides on the it fundamentally leans towards being a conservative one. I think, though, that there are more sophisticated algorithms here that are possible that would be a little bit more global in nature. So for example, you could use extra information to try to correct the barcode a little bit harder. One piece of information we don't use is like the quality scores for the bases. But even beyond that, you could ask questions like, okay, maybe I want to, for the purposes of the next phase of my algorithm, allow this read to be assigned to either of the cells. And I am going to determine the ultimate assignment of the barcode based on some global parsimonious solution. If including this read allows one cell or the other to find a more parsimonious solution, then I'm going to allocate that read to that cell. I think these are really interesting questions, but we have not gotten there yet. But I think that the short answer to that question is, the whitelist or the permit list barcodes that are given to us are what they are. They're generated by 10x Genomics, and a lot of them are close together. And so we're just doing the most efficient and conservative thing we can to try to avoid ambiguous assignment. Hopefully that answers the question.
Hey, great. Unless there's any more questions in the chat, I'd love to thank you again, both of you, for presenting today. I think both were really interesting.
Absolutely. It was my pleasure. Thank you very much.
And thank you everyone for joining us today.
Thanks, Rob.
Thank you. Take care. Hope to see you soon.
Sure. See you as well. Bye.